Our final speaker for today is Vaagmi Shukla, a junior from Hopkinton High School, and she's going to introduce to us the big world of gene therapy and all of its possibilities. Please welcome Vaagmi.
Hello, everyone. Have you ever wondered what it would take to fix a disease? Not just treat the symptoms, but to actually repair it at its root cause. Imagine a technology so small that it's invisible to the human eye, yet powerful enough to change lives forever. That's what I'm here to talk about tonight. Thank you, Sahasra and Teju, for introducing me. I am Vaagmi, and I'm a junior at Hopkinton High School. I have research experience in the field of neuroscience, and I would love to share about AAV gene therapy focus as a high schooler. We're going to dive into AAV therapy and innovation at the intersection of neurology and gene therapy. AAV gene therapy is revolutionizing the way that we understand neurological disorders. I'll share the science, unravel some of the myths, and most importantly, show how this small vector is going to give us hope and give millions of people of hope living with debilitating neurological conditions. Let's begin. I want to start by discussing what is gene therapy. Gene therapy is think of the human body as a highly complex machine. Sometimes the machine malfunctions because it's damaged or it's missing parts, which are our genes. Gene therapy aims to fix these malfunctions by repairing, replacing, or modifying faulty genes at their source. AAV, or adeno-associated virus, plays a pivotal role in this process. First discovered in 1965 in collected cultures from rhesus monkey kidney cells, AAV contains a single-stranded DNA genome in which therapeutic cargo sequences within certain size limits can be fitted within the flanking of inverted terminal repeats. To suffice, these are harmless viruses that have been re-engineered to serve as microscopic delivery vehicles of medicine. Imagine them as tiny drones carrying healthy versions of genes into cells to replace the broken ones. This potential of technology goes for beyond just treating symptoms. It's about restoring lost function, whether that means enabling someone who has a rare disease to walk again, or to give a child born with blindness the gift of sight. AAV is rewriting what is possible in medicine. Next, I want to discuss the science behind AAV gene therapy. Let's break this down further. AAV vectors are like Trojan horses, as we know, but for good. In their natural state, these viruses are harmless to humans. Scientists have engineered them of removing their harmful contents and then leaving behind an empty shell called a capsid. We know that viruses are, viral. Therefore, what we do is that we get rid of these viral genes and then replace them with the desired gene. So after this, viruses are able to make copies of themselves rapidly throughout our genome, and this is what is the key to AAV gene therapy. This shell is then loaded with a healthy copy of the desired gene that needs to be delivered. Once ejected, the AAV vector enters the target cells into the cargo, and this contains the correct genetic instructions to repair the error. So what makes AAV gene therapy so special? It is small, it is versatile, and it can be tailored to target specific tissues, such as the brain, liver, or retina. These affinities are called serotypes. For example, AAV9 is particularly effective in the brain. This is because it has an affinity to get through the blood-brain barrier easier. And so with its ability to pass through the blood-brain barrier easier, it's able to enter the brain easier. While AAV2 is effective for eye or, the retina-related diseases. This precision makes it a game changer for diseases that were previously considered untreatable. Next up, I have an illustration of what the AAV vector typically looks like. And as you can see that there is a capsid, and inside the capsid we have the desired gene. And of course, there are three main pros of AAV therapy. Safety, they don't cause disease in humans. Efficiency, they can efficiently deliver DNA to a wide range of tissues, including neurons. And longevity, once inside the neuron, AAV vectors can persist for long periods of time without integrating into the host genome, reducing risks of unintended genetic changes. AAV vectors are very precise, as we know, and so they can be applied to different diseases. For example, Huntington's disease. For Huntington's disease, where a mutated gene produces a toxic protein, AAV vectors can deliver gene silencing instructions to solve the disease at its source. Another example of this is SMA, spinal muscular atrophy, in which a single dose of AAV therapy can replace a missing gene and allow the neurons to thrive and enable them to regain muscle control. Next up, some of you may heard of CRISPR, the revolutionary gene editing tool that can cut DNA to remove or correct errors. Think of it as a highly precise pair of molecular scissors. But here's where AAV therapy comes in. While CRISPR is great for editing, AAV vectors can carry the correct DNA sequence and deliver it to the right cells. So together, they form a powerful toolkit capable of curing diseases at their genetic root. For example, in diseases like sickle cell anemia, CRISPR can fix the mutation, and AAV can deliver the healthy instructions to ensure that new cells are created without the defect. This kind of collaboration is what makes the future of medicine so exciting. So, why neurology? The stakes are high. Because the stakes are incredibly high, in fact. Around 1 in 6 people globally suffer from a neurological disorder, whether it's Alzheimer's, Parkinson's, epilepsy, dementia, ALS. Neurological diseases don't only affect physical health, they touch the core of who we are. Memory, movement, speech, even personality, everything that makes us human and ourselves is at risk. Traditional treatments like medicine or surgery exist, and they can help manage symptoms. But they rarely address the root cause. That's where AAV therapy offers hope. By targeting the genetic underpinnings of these diseases, we can potentially restore function and in some cases, reverse the damage entirely. So let's debunk some misconceptions. There are some myths such as we are playing God. But the fact is, AAV targets specific genetic errors. It doesn't alter your personality or your free will. And another myth is that brain therapy creates superhumans. But the fact is, it heals damaged brains, not enhances them. So there are a lot of pop culture and popular myths regarding therapies like these. Movies often exaggerate brain science. Think of Lucy and the claim that humans only use 10% of their brains, or Limitless, where a pill unlocks infinite intelligence. In reality, gene therapy isn't creating superhuman brains. It's healing damaged ones. So let's discuss applications of AAV gene therapy. So, as I mentioned, in the retina, it can restore vision for individuals with inherited blindness. In the brain, it holds promise for treating epilepsy and other neurological diseases. And it's also being used to target the liver for genetic diseases like hemophilia. What's incredible is the precision. So you can target a specific part of your body, which is so fascinating. Each application moves us closer to a world where no disease is beyond our reach. Manufacturing and delivery. You may be wondering, so how does this actually get in my system? How does this enter my genome? Creating AAV therapy is a highly sophisticated process. It starts with designing the vector to target specific tissues, and whether it's the brain, the liver, etc. Next, the vector is produced and it is purified in order to ensure that it's safe and effective. Finally, the therapy is delivered to the patient, often by injection or infusion, and each step requires a meticulous precision. but the result is worth it. The therapy is capable of changing lives. So let's discuss the future of AAV therapy. It is truly limitless, and beyond genetic disorders, scientists and researchers are exploring its use in cancer immunotherapy, vaccine delivery, and even non-genetic diseases. Imagine a world where AAV vectors are as common as antibiotics, a world where diseases we once thought were nothing more than historical footnotes. That is the future that we're working on. So, if you could fix one neurological disorder, epilepsy, Parkinson's, ALS, what would it be? I'll give you some time to think, and we can share. Or rather, what is a disease that you're wondering how it may be cured using an AAV? Anyone? No? We can discuss a very common disease that was actually mentioned in this TED Talk. So, Parkinson's disease. It is caused by the death of dopamine-producing neurons in the brain. Currently, treatments like levodopa help manage symptoms, but they do not address the underlying genetic cause. Gene therapy can deliver a gene to the brain that encodes for GAD, glutamic acid decarboxylase, an enzyme that helps produce dopamine. By delivering the gene using AAV vectors, researchers aim to restore dopamine levels in the brain, which improve motor function and help Parkinson's patients. By using AAV vectors to deliver a gene encoding GAD, researchers are attempting to replace the missing dopamine in Parkinson's patients. This has the potential to significantly improve motor function and quality of life, addressing the root cause rather than masking the symptoms. Another example is spinal muscular atrophy, as I mentioned earlier, SMA. SMA is caused by a mutation in the SMN1 gene, which is essentially for the survival of motor neurons. Without the functional SMN1 gene, motor neurons deteriorate, means that they break down, leading to muscle weakness and atrophy. In a breakthrough treatment, AAV vectors have been used to deliver a functional gene of the SM1 protein gene to motor neurons, halting the progression of the disease and in some cases even reversing the damage. In SMA, a single dose of gene therapy using AAV vectors that can deliver the missing SMN1 genes to motor neurons, which helps prevent the degeneration of muscle cells. This therapy has shown remarkable success in halting and even reversing the disease's effects, giving children with SMA the chance to walk and breathe independently. Last up, I also want to address some challenges with AAV gene therapy. There are size limitations. AAV vectors can only carry small genes, which is less than 5,000 base pairs, and larger genes or more complex therapies may require other delivery methods. Also, there is the immune response. After all, you are reusing a virus in order to get this inside your genome. So, some patients may develop immune responses to AAV vectors, especially after repeated doses. These can hinder the effectiveness of the treatment. Lastly, targeting precision. While AAV vectors are effective in targeting specific areas, there is still room for improvement in precision targeting throughout the body. While AAV gene therapy is showing incredible potential, there are challenges to overcome. Ultimately, I leave you with this statement: gene therapy through the use of AAV vectors represents one of the most promising frontiers in neurology. These tiny vectors are small, but they are mighty, and they are not just delivering genes, they are delivering hope. By correcting genetic errors in the brain, we are on the verge of treating, perhaps even curing, some of the most challenging neurological diseases of our time. Thank you.